Hi guys, welcome to today's video. We are going to look at some NCEA Level 1 science, looking at the achievement standard of genetics variation. So as a topic, as a title suggests, we're going to look at Punnett Squares and Pedigree Charts today. But just before we do that, we're just going to quickly recap on some of the key ideas so that when we refer to certain terminologies, it will make sense. Otherwise, you just doesn't understand what we're trying to talk about here. So just really quickly, some of the key things that you need to some of the key definitions you need to understand. Everything. So we started looking at DNA, and then so what is a DNA? You can look up on, let's say, I've given my class some definitions, and this very popular question like what is a DNA? The double helix structure that contains all the genetic information, and then from your DNAs, you are in one section of the DNA. Is called a gene. One gene represents one particular trait, one physical characteristic, but then out of that one trait you have different versions. You have different versions of the same trait, different versions, and these are coded by what we call alleles. Okay, so when you have different versions, why do you have different versions? Because we have different genetic the different base sequencing. So that's what that is, different base sequencing. What different base sequence, and what is base sequence? That's where the ATCG stuff comes in. So if we break it down to short, so all of us have DNA in our cells, and then one section and the DNA is really big, really long, and if you look at one particular section, that is what we call a gene, and one gene codes for one characteristic, one physical trait, but then there are different versions of that particular trait. Like, say, for example, if you look yourself in the mirror, you look at your hairline, there's a very unique trait called widow's peak. So it's like a V-shaped hairline. A lot of people's hairline is just straight. A lot of us, like I have a V-shaped hairline. So that's different versions of the same trait. Why is that? Because for that particular same gene, we have different alleles. Okay, so why are they different? Because for the same gene, they have different base sequence. Like, say, for example, one could be A, T, C, G, TA, for example. This could be one particular allele. For the other one could be G, C, T, A, TA, for example. And both of these base sequence, they code for the same gene, but as you can see, they're different, so they result in a different protein, and that results in a different physical appearance, which is a physical trait. Okay, now alleles. So we use capital letters for the dominant allele. So the dominant allele. So because it normally, to simplify things, we say there are two versions of each gene. So the one that's more dominant, that's always being displayed, is what we call, we assign that with a capital letter, and the one with the lowercase is going to be the recessive allele. Now, the recessive allele is only going to display if there's no dominant allele present. Okay, so that comes down to, like, say, for example, if I use, capital H, lowercase h, there's three different combinations of how it could look like. So the H could mean, the hairline example, H for hairline. So you could have three scenarios. This, when you have one of each, a capital H, lowercase h, this is what we call a heterozygous. This is a heterozygous genotype. And when you have two capital H's, this is homozygous dominant. Homo means the same, so homozygous dominant means I have two dominants. Two slow H's, this is also the same, so it's homozygous recessive, because I have got two recessives. Now, just remember, with the heterozygous, you don't need to say heterozygous dominant or heterozygous recessive. Hetero just means different, so heterozygous means one of each. Okay, so I'm gonna just quickly recap these because we're gonna need to use these for later. Okay, let's look at some particular questions. Okay, so the best way to do genetics is by looking at some questions, especially with Punnett Squares and Pedigree Charts. So let's have a look at these. Okay, so we're looking at rats in this particular example. Let's highlight the important part. Albinism is caused by a recessive allele small a. So when you get told this, and they always tell you they always give you the letters to use. They always tell you which one's positive, which one's dominant, which one's recessive. Some questions they give you the pedigree chart and ask you to figure out which one's dominant, which is recessive, which we can look at in some of the examples that we got given below. Okay, so let's just really quickly link back to what we done just now. So if we have an albino rat and we know it's that particular genotype, the allele that codes for that is a small a. So if you have an albino rat, so that means the genotype of this particular rat must be small a. Okay, because there's no other scenario. Because as soon as you have a capital A in the genotype, the rat will be black, will be normal. Okay, so that means this is going to be small a. When you have a heterozygous black rat, so heterozygous means one of each, so you have a capital A, lowercase a. So why does a rat appear this particular color, like a normal rat that you see, is because of the capital A that you get given in the in the question. So this is a very messy Punnett square. So this is what they so in terms of like, this is an older version of some of the exams, but this is what you kind of get given nowadays. They do it like this. Okay, so this is what it kind of looks like nowadays. So it's the same, exactly the same thing, but these type of table questions much less common now. They just give you the Punnett square. So how do you do Punnett square? You literally just identify the two alleles and put them one each into the columns, and then you simply combine them. Like, say for example, this and this goes here, so it's a capital A, lowercase a. This and this goes here, so this is a capital A, lowercase a. So this is small a. So if we come back to this question, it's literally like, all right, so if I combine this a, and if we combine with this a, so this is capital A, lowercase a. If you combine this a with this a, this is lowercase a. If you combine this one with this one, this is capital A, lowercase a, and this is lowercase a. So you get this exactly the same outcome. And just to represent a Punnett square, so what do we use a Punnett square for? A Punnett square is simply used to predict the genotype, the possibility of getting different genotypes in the outcomes of these two organisms having offsprings, and we can predict the possibility, what are the chances of the baby rats are going to be normal color or albino, and we can figure out a percentage. So if we look at this particular question, this is going to be, if you look at that, so these are my 4 outcomes. I have got 2 of them. I got 2 big A, small a's, which is heterozygous. I got 2 small a's, which is homozygous recessive. So out of 4, I have 50%, 2 out of 4, going to be big A, small a, which means they are going to be a normal black color. If I get 50% small a, which is homozygous recessive, that means I'm going to get an albino rat. Okay, so does that mean, like say for example, if these rats were to reproduce and have 4 baby rats, are they all going to be 50-50? Obviously not. It's just every single time a baby rat, an offspring is being reproduced, there's a 50% chance. It's like a coin toss, like with humans, like boy or girl. It's just a flip of a coin, and it just depends on what the coin turns out to be every single time. So in our school we have I've known siblings with like a very big family, 6-7 sisters, no brothers in the family, probably only actually there's one brother at the end, the youngest one's a brother. So it's just a coin toss every time, and just happen to be girls for like the first 5-6 times. Okay, so that's unlikely, but it can happen. Okay, so this is the first question that I thought I would look at. Now this is looking at the pedigree chart. This is looking at so we need to remember albino is small a, and then so albino rat is small a. This is from the previous question, and normal is just capital A. All right, so just remember that. Now the albino rats, well, so this is your typical what do you call it, pedigree charts. Okay, so you look at the family tree of the physical traits that we're looking at, and then you can actually predict what the genotypes are of the following rats. We can do better. We can do, instead of doing 4 and 6, let's do all of them. Okay, let's do all of them. It's good practice, and that's often how I teach my guys when we do pedigree charts. Don't just look at the question. If there's a pedigree chart, you have the clue in front of you. Can you actually figure out all of the answers? All right. So let's come back to the example. So albino rats are small a's. Okay, so that means this one must be small a, small a. This one must be homozygous recessive. This one must be homozygous recessive, because they have to have a lowercase a and another lowercase a, because if they were to have the dominant capital A, they will be normal. Okay, now all of the other guys, like say 2, 3, 5, 6, you know, so on and so forth, all of them have to have at least one capital A's. Okay, so you can just literally do that, go above them and write a capital A in front above them, because they have to have at least one. Okay, so it doesn't matter what the other allele is, as long as they got one capital A, that means they would display this particular phenotype, which is the normal hair color. So how do we decide what the other thing is? So this is where you need to look. I'm just gonna clean this a little bit, because you get the idea, all right? Otherwise, I just want to clean it out. Otherwise, it's gonna look really messy. Okay, so let's ignore these. All right, let's use a different color. So let's look at the offspring of number 1 and number 2. So number 1 and number 2 have 3 offsprings, which is this guy, and this guy. Okay, so let's look at the Punnett squares. Now, if we were to, you know, I don't want to write long explanations, I just want you to understand this. So can you see, so this has to have a capital A, and the other letter has to be a small a. And you probably go, how do you know that? How do you know it's not a capital A? Now, how do you know it's not homozygous dominant? Well, the species of interest is going to be this guy. The organism of interest is going to be that guy, because these two parents, you have this fella and this fella, they managed to have 3 offsprings, and one of them is a small a. So the only possibility that you can get a small a is that you got one small a from mom, and then you got one small a from dad. Okay, so that means this doesn't tell us the gender, but this parent, this could be mommy rat or baby daddy rat, doesn't really matter. It has to have a small a, because otherwise, where would the second small a come from? You can't remember, from when we inherit alleles from the parents, you get half from each parent. So that means you get half of capital A, small a, so which means you guarantee a small a from this particular rat, but then you have to get the other a from this rat. So that's why this number 2, sorry, it's going to be a bit messy again. Number 2 has to be heterozygous, because there's no other option, there's no other possibility for this number 4 to inherit this small a. Okay, so it has to have a capital A. So let's do the Punnett square. If we know it's a capital A, small a, then you can see that this is going to be a heterozygous, homozygous recessive, and that explains it. Okay, and then you can even prove it. If I do big A, small a, can you see that there's absolute no chance that you get an albino rat out in the outcome? Absolutely no chance. Okay, so by doing that, we figured out all of the ones above. So we know this is small a, this is big A, small a, this is small a, this is number 5 is big A, small a, number 6 is big a, small a, because the only possibility for the offspring to have a normal hair color is because they got the capital A from, you know, number 2, and then they got the other small a from number 1. Okay, so we figured out 5 and 6. So if you actually look at this, number 4, number 6, we actually have finished the question. Now let's do a little bit better. Let's do more, because it's all about understanding. You're not here just to get if you want more answers, just go onto the NCEA website and read that. But that's not what you're here for. You're here for the understanding, because you can always memorize how to write, but then especially during lockdowns, you probably want someone to explain to you. All right, let's look at number 7. If you look at number 7, so again, number 6 and number 7 want to reproduce number 12 and number 13. Now number 12, again, number 12 is small a, small a. So that means number 6 has to have a small a, and number 7 has to have a small a. But we know number 7 is a normal hair color, so it also has to have a capital A. So it's to do with logic. It's like, I love doing pedigree charts. It's like solving puzzles, and you can't just look top down. You have to sometimes look backwards from bottom up to determine what type of genotypes we're looking at. Okay, so number 7 is capital A, small a. Now for number 13, we actually don't know, because if you do the Punnett squares of these two parents, oops, you can see there are 3 possible genotypes. So the ones highlighted that I'm highlighting right now, they will all can become number 13, which can be the genotype. So it could be capital A, or capital A, small a. So either homozygous dominant or heterozygous, we don't know. And how could we know? Get that rat, select that rat, and then get it to reproduce, and then look at the future generations. Okay, now if you look at number 3, we don't know. It could be the capital A, it could be the capital A, small a. Because if you look at 8, 9, 10, 11, these guys would have to be capital A's, small a's, capital A, small a, capital A, small a, and capital A, small a. And then you go, how do you know that? We look at the parents. We look at number 4 has to be big small a, and remember these number 8, 9, 10, 11, they have to inherit 1 allele from each parent. So that means it's 100% chance you get a small a from the number 4 rat, and then you got a big A from the number 3 rat. Okay, because why do you get that? Because they look normal. Okay, so they have to have a capital A. So this is why they have to have a capital A and they have to have a small a. Just using logics. Okay, now look at this. So rat 3 was not an offspring, so we're looking at rat number 3. So this is we kind of jumped ahead with the question. So if we look at the chart here, let me just rub this out. If we look at the chart here, and then we go, all right, what is possibility number 3? So we know number 4. So we know number 4, we know rat number 4 has to be small a. We know rat number 3 could be big A, small a, or big A, big A. Because all of their children, all offsprings, all of their offsprings are big A, small a, with the reasons that we talked about here. So if you look at the question, state the possible genotype. It could be this or could be this. Why? Because it's normal color, so that means it has to have a dominant trait, and we don't know what the other one is, because if we can't look at its genotype, we can only look at the physical structure. You explain why both genotypes are possible, because if you actually look at the possibility, so these are the 2 possibilities, and then they have to mate with the small a, and then if you actually complete the Punnett squares, you can see there is so for this one, there's 100% chance big A, small a. And what does that mean? So this is a genotype, and the phenotype is their hair color, which is the normal black color. And then if you look at this is 50% genotype is going to be 2 out of 4 going to be big A, small a. 50% is going to be small a, small a. Okay, so that means there's a 50% chance they'll be normal, but then there's a 50% chance they are albino. Okay, so which one, but one is more likely, so this is more likely, so that means it's more likely for number 3 to be capital A, which is homozygous dominant, but it is still possible for rat number 3 to be heterozygous, because like I just mentioned with the examples, it could be, if you know any family or friends they have siblings, and 3 or 4 siblings, and they're all of the same gender, it could just be by chance, and it has happened in this case as well. Yes, it could be a heterozygous rat, but then just by chance, 50% of the time, boom, 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 hit it 4 times in a row, and then you got 4 rats and a normal. So what we could do to be more certain about the genotype of rat number 3, get it to keep mating with rat number 4. Okay, because the moment you see that one of them is one of them is albino, boom, you know that the parent is heterozygous. Okay, and if you after like 5 more rats, 10 more rats, baby rats that were born, and there's still no albino rats, then it's very likely that it's going to be capital A. Okay, homozygous dominant. So it's to do with probability, and that's why it is important. If you're interested in taking biology, statistics is often a very, very important subject to take alongside biology, because you get a lot of data and you need to analyze the data, and stats do teach you really well in that regard. Okay, so let's look at a few more. So this is another condition. So this is to do with horses having a white patch on the head, on the body of horses, very, very common. So in horses, let's look at this. Bipodism is a dominant trait, which is capital H. Normal is recessive. Okay, so let's remember that. Now, when we look at the chart, normal is shaded, and then these guys are, not filled in. So that means which one's recessive? The normal color is recessive. So that means normal is recessive. So don't even think about it. Just go small h, 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 small h. And this is how you can analyze. You can probably see I don't even have the questions. Like, this is just a pedigree chart. I don't even know what the question is asking you to do. But if you can do these steps, you can answer any question they give you, because you already solved all the species genotypes. And anything else, you would have answered any possible question the examiner could have thrown at you. Okay, so just remember, look for the recessive trait, look for the recessive trait here, and then you can literally just put homozygous recessive, the small letters for these ones here. So let's decide number 1. Number 1 must be heterozygous. Why is that? Because all of the children are small h's. Okay, so that means it has to have. Now, because it's the pipode, so that means the pipode, so they have to have a capital H, then it has to have a lowercase h, because this gives you, that's given from here, and the small h's from 5, 6, 7, 8 tells you that how did they get the small h? They inherited from both parents. Okay, so that's why number 1 is capital H, lowercase h. If you look at number 3, number 4, so same thing. This has to have a capital H. This has to have a capital H. Yeah, so regardless of what the other one is, they have to have at least one capital letter, because they are showing the dominant trait, because they told you they are pipode, and that's why they have to have at least one capital H's. And then you can look at the children. Number 10, number 11, number 12 are both small h's. So what does that mean? They would have inherited the small h from each parent. Okay, so that means you get one of the small h's from each parent. That's why number 3 and number 4 are heterozygous. Okay, so if you look at number 9, number 9 is hard to decide, but these offsprings here, they have to be capital H, lowercase h, capital, they have to be heterozygous. Why is that? Because number 8 is normal, so it has to have small h, small h. So this is where the small h come from, because you remember you inherit one allele from each parent, so they have to get the h from number 8, the small h, because there's no other option. You're 100% going to get that. Number 9 is going to be, it's going to be capital H plus something else. Could be H, could be H, capital H heterozygous, could be homozygous dominant, but it doesn't matter, because these offsprings are pipode, so that means they would have inherited the positive, the big H. Okay, so there you go. That's how you can answer the questions. Let's do one more before we wrap it up. Photic sneezing. So we're not interested in what the condition is. We're looking at this. The sneezing is dominant to unaffected. Okay, so just look here. So what's unaffected? I always go with the recessive trait because that's going to make your life a lot easier. Unaffected is small a. Unaffected are these guys. So straight away, small a, you know, these things are these people are all going to be homozygous recessive. Okay, regardless. And if we apply the same thinking, if we got number 5, 6, 7 all being homozygous recessive, that means that means their parents, again, if you got 2 small a's, that means their parents must have a small a each. So we got the small a's from this parent, then that means we have to have the small a from number 2. Okay, so you know they have to have one big capital letters each because they are displaying that trait, and we can just figure out what the other one is. And then if you look at number 3, number 4, if you look at the children, number 8 is showing small a, so same thing, would have got this from one of each parent, so that means number 4 will be heterozygous as well. And that means this is going to be small heterozygous, heterozygous, heterozygous. And you may go, how do you know that? Because if you look at, you know, who are they children of? Number 9, 10, 11 are children of number 3 and number 4. And then you can, you know, work backwards. You can, you know, this is how they have to inherit the small a, one small a from parent number 3, and then because they are affected, so they would have got the dominant trait as well. Okay, so number 12 is also, so if you look at number 11, number 12, number 11 we know is going to be big A, small a. Number 12 is going to be capital A for sure, but we don't know what the other one is. But then if you look at number 14, number 14 has capital small a, small a. That means you would have got a small a from each parent. So this is not a question mark. This is going to be heterozygous. Okay, so hopefully it does make sense. It does take quite a bit of practice. That's why it's actually good that you're watching these, because you can always rewatch the explanation. And the best way to have a go at it is to go through the past year's examination papers, which I'm aiming to finish relatively soon. But you have the 2018-2019 exams available on my YouTube channel, so feel free to browse through the playlist and find the particular exams, and just make sure that you can produce something similar in November or December. I can't remember when the exams are. Okay, so hopefully this has been helpful, and look after yourself, and I'll see you guys next time. Bye bye.